Welcome back to the Deep Dive. Today we are taking a massive stack of research papers on microbiology, neuroscience, and even oncology. And we're tracing the story of one single tiny molecule that connects them all. If you've ever seen the movie Lucy, you might remember the fictional drug CPH4.
Oh, right.
The one that's supposedly unlocked, 99% of the human brain's potential, granting superpowers, omniscience.
The ultimate brain hack. And this is where reality gets, just as strange as science fiction.
Go on.
CPH4, the pathway around it, 6-carboxy-5,677-8 tetrahydropterin, is absolutely real.
Way.
It's not a miracle drug, but it is a crucial precursor molecule made by certain bacteria, like the ones living in your gut right now.
So our mission today is to follow that real bacterial molecule, CPH4, all the way to its final form, a hypermodified nucleoside called Q-azine, or just Q.
Right.
We are going to explore how this single modification, which we can't even make ourselves, is salvaged from our microbiome.
Yeah.
And how it literally controls the efficiency and the accuracy of our most foundational biological process-making proteins.
Exactly. We're diving deep into the microscopic editor that dictates everything from a bacteria's ability to shrug off antibiotics to how our neurons fire and how our memories are laid down.
It's an incredible link, then, between our microbial neighbors and our own neurological health.
A completely systemic link.
Okay, so let's unpack this. We've said Q-azine, or Q, is a hypermodified nucleoside, but where does it actually live?
It lives in transfer RNA, or tRNA, specifically at position 34, which scientists call the wobble position.
For listeners who maybe don't think about RNA on the weekend, what is tRNA's job, and why is that wobble position so critical?
Okay, so think of the ribosome as the protein factory. The mRNA is the blueprint.
And the tRNA is the translator, the delivery truck, maybe?
Exactly, it's the delivery truck. It matches the code on the blueprint, the codons, to the right building blocks, the amino acids.
Okay, so where does the wobble position fit in?
The wobble position is like the flexible hitch on the back of that delivery truck.
Yeah.
It allows that tRNA to recognize and latch onto several slightly different versions of the code.
So it gives the system some efficiency, a little bit of flexibility.
Little margin for error, yeah. Now, Q comes in and modifies that flexible hitch. It fundamentally regulates the efficiency and the fidelity, the accuracy of the whole process.
So Q is like the quality control officer.
A quality control officer making sure key amino acids like tyrosine, histidine, and aspartic acid get slotted in correctly and at just the right speed.
So it's not just about accuracy, it's also about the pacing of the whole assembly line.
Precisely. And here is the profound twist, the part that forces us to look at the microbiome.
Right.
Eukaryotes, so every cell in your body. We can't synthesize Q from scratch. We are completely reliant on salvaging the precursor, which is called Qin, or Q.
Which means we get it from our food, or, more importantly, our gut bacteria produce it for us. The same bacteria making that CPH4 precursor.
That's the one.
But how does the cell actually do that? How does it perform the swap?
That's the salvage pathway. It's carried out by a microscopic construction crew called tRNA guanine transglycosylase, or TGT. It's a complex machine with two parts, QTRT1 and QTRT2. And it literally performs a surgical switch. It cuts out a standard guanine base from the tRNA and snaps in the salvaged Q-ine base.
So it's a specialized recycling service, but it's using parts supplied by our gut bacteria.
Perfectly put.
But if we're that reliant on the gut for this component, doesn't that make us incredibly vulnerable? I mean, what happens if that microbial supply chain gets disrupted?
It's a great question, and the answer is that the system is incredibly sensitive. Q modification isn't a simple on-off switch. It's more like a sophisticated tuning dial.
Okay.
For instance, take Vibrio cholerae, the bacterium that causes cholera. When you take Q out of its system, misdecoding by one type of tRNAtor at ASP codons increases.
Okay, that makes sense. You lose quality control, you get more errors.
But, and this is the counterintuitive part, the loss of Q decreased misdecoding by a different tRNase at another codon.
So it's not universally reducing error.
No, it's highly specific. It's creating an optimal balance, sometimes reducing error, sometimes accelerating things to keep the cell in equilibrium. When that tuning dial is broken, the fidelity of the entire system suffers, but in these really complex, nuanced ways.
The complexity is amazing. Now let's go back to the source in the bacteria and talk about the enzymes making CPH4. What does that initial pathway look like?
It's a marvel of chemical engineering. In bacteria, you have an enzyme, queef, performing a crucial reduction step. Then you have Qui, and that's the enzyme that actually produces CPH4 BH4 from its starting fuel, H2NTP.
And what's fascinating here is the structural economy. These enzymes belong to the tunnel fold or T-fold superfamily. And a fold is the basic 3D architecture of the protein, right?
Right, the architecture.
So why is it so remarkable that 7 of these enzymes share the exact same architecture but do wildly different jobs?
It speaks to evolution's ingenuity. The T-fold is an ancient, reusable blueprint. These enzymes all have the same tunnel-like structure, but tiny tweaks to the active site, the specific amino acids inside that tunnel, allow them to perform completely diverse chemistry.
So it's catalytic promiscuity.
In action, a common scaffold that evolution has specialized over millennia to create new functions.
It's like building 7 totally different vehicles, a sedan, a truck, a race car, all on the same foundational chassis.
Exactly. And we see the structural similarity causing some functional confusion even between bacteria and us. Compare the bacterial CpH4 maker, QD, with our own mammalian version, MPTPS.
And our MPTPS is vital. It makes BH4 tetrahydrobiopterin, which we need to synthesize key neurotransmitters like dopamine and serotonin.
Precisely. Both CreD in bacteria and MPTPS in us start with the exact same material, H2NTP, but one is highly dedicated to making BH4 for our brain, and the other is dedicated to making CpH4 for this Q pathway.
But there's an overlap.
There is. If you overexpress our MPTPS in a mutant bacterium that can't make its own CpH4, our mammalian enzyme can actually step in and partially do the job.
Wait, really? So our brain chemical factory worker can moonlight as a bacterial component producer?
It can, but it's very, very slow. The structural similarity is there, but the rate is vastly slower because it's not optimized for that reaction.
And what's the difference?
It often boils down to a single histidine, a tiny pair of amino acids that the bacterial QD has. It acts like a turbocharger, dramatically amplifying the rate of CpH4 production.
That's brilliant. Minor evolutionary tweaks dictating major functional differences. Now let's switch from ancient blueprints to modern microbial warfare. How do bacteria use Q dynamically in their understanding?
The Q modification levels are not static at all, they're exponential. When bacteria sense stress, they change the level of Q on their tRNAs.
Okay.
In Vibrio cholerae, for instance, tolerance to aminoglycoside antibiotics is directly linked to increased expression of the TGT gene, the one that does the Q modification swap.
So more Q helps them survive the antibiotic.
It seems to. Higher Q levels appear to help the bacteria survive the attack by changing how fast and accurately they decode certain tyrosine codons.
So Q is basically helping the bacteria adjust its translation pace to survive a chemical onslaught. It's an internal adaptive mechanism.
Absolutely. And the effects go even deeper. A study on Q-deficient E. coli, where they knocked out the TGD enzyme, showed the bacteria acted as if they were under massive oxidative stress.
Just from lacking Q.
Just from that. They became hypersensitive to stressors like hydrogen peroxide. A lack of Q basically leaves the bacteria naked and vulnerable.
And it messes with essential supplies too, right?
It does. That same Q-deficient E. coli showed disturbed metal homeostasis. The main nickel import system, the Nic ABCDE operon, was repressed. So even when nickel was around, the bacteria couldn't point in efficiently.
And then there's the completely unexpected role of another Q pathway enzyme, Q, in literally stopping cell division.
This is a fantastic example of a protein being repurposed for crisis management. When E. coli is attacked by antimicrobial peptides, which our own immune system uses, it activates a big stress signaling system.
And that system.
That activation dramatically increases the expression of Qui, and high levels of Qui then act as a cell division block.
Meaning the bacteria can't reproduce.
They try, but they can't finish. They start to filament, they just stretch out and get longer without dividing. Qui physically goes to the division septum, the exact spot where the cell is trying to pinch off.
So if you get rid of Qui,
if you delete the Q gene, you suppress that filamentation. It proves its surprising role. The Q pathway chemistry is being used to tell the bacteria, hey, it's too dangerous to reproduce right now. Shut down the division line.
incredible. A molecule for quality control and translation moonlights as an emergency brake on reproduction. Which brings us full circle back to the microbiome and how this microbial compound dictates our health specifically in the brain.
And this is where that BH4 link becomes critically important for us. When Q levels are deficient, it compromises the overall production of tyrosine.
And tyrosine, as you said, is needed to make BH4 the cofactor for dopamine and serotonin.
Right. So if the Q supply from the gut drops, you have a direct pathway to reduce BH4, potentially leading to a crash in crucial neurotransmitter levels.
A crash like that is immediately relevant to conditions like Parkinson's disease and even metabolic disorders. disruptors like PKU. It's the ultimate example of the gut-brain axis at work.
And we have compelling evidence that we might be able to intervene. A synthetic version of QN, called STL-101, has shown neuroprotective potential in lab models.
What did it do?
It decreased phosphorylated alpha-synuclein, a hallmark of diseases like Parkinson's, and it protected dopaminergic neurons from specific toxic injury.
So synthetic Q can act as a shield, at least in a petri dish.
Exactly. The research also highlights Q's foundational role in mitochondrial health, which is a common denominator in almost all neurodegenerative diseases.
How so?
Q seems absolutely essential for mitochondrial translation quality control. When Q is depleted, cells' power plants start to break down. You see increased proton leak, decreased ATP production.
If the cell's power grid is faulty because of poor quality control and translation, that's a direct link to the major drivers of Alzheimer's and Parkinson's.
It ties the whole picture together. But maybe the most surprising finding in all the neuroscience data is the role Q plays in sex-dependent cognition. This is a game changer.
Okay, tell us about the QDIRT1 knockout mice. What happened when they were deprived of Q?
They found profound sex-dependent defects in learning and memory. And crucially, the female knockout mice showed a notable reduction in learning capacity and memory retention compared to the males.
So they couldn't remember spatial information as well.
Not nearly as well. We are talking about one single molecular deficiency resulting in a gendered cognitive defect.
Wow. And this correlates with human data, doesn't it? I think the sources mentioned that plasma QN levels are often naturally higher in women.
It absolutely raises a fascinating hypothesis about why neurodegenerative diseases often show a sex bias. If Q levels are a critical determinant, and if the baseline supply or usage differs between sexes, that could be a long-term factor.
So let's zoom back in on the core mechanical failure behind this. We've used the term traffic jam before. Explain how losing Q actually causes this systemic chaos on the ribosome.
That analogy is perfect. Losing the Q modification creates an overall imbalance in translation speed. Imagine a highway where some lanes, the AU-rich codons, slow to a crawl while others speed up. It causes a massive pileup.
A systemic slowdown.
A systemic slowdown caused by congestion and stalling at those few crucial Q-decoded codons.
It's the butterfly effect. A single missing modification causes the entire translation machinery to glitch globally, and that glitch shows up as a complex memory defect in a sex-specific way.
It connects molecular detail to behavioral outcome in the most elegant yet terrifying way.
Finally, let's talk about the clinical horizon. This molecule is essential for our brain health, but it has a dual nature, especially when we look at cancer.
We circle back to that salvage enzyme, QTRT1. That's the part that actually inserts QN into the tRNA. In studies on breast cancer cells, knocking out QTRT1 caused a significant reduction in tumor growth, volume, and proliferation in mouse models.
Hold on. The same molecular machinery that is vital for preventing neurodegeneration is also being used by cancer cells to fuel their growth.
That's the profound paradox.
That's the ultimate dilemma.
It is. It suggests that Q modification provides a growth advantage that aggressive, rapidly dividing cells like tumor cells exploit. This modification supports proliferation. Q isn't just a nutrient, it's a foundational resource that the host has to manage carefully.
So this whole deep dive has taken us from the fictional CPH4 to a real-life metabolic linchpin Q-acine. We started with an ancient, promiscuous bacterial enzyme, followed its precursor through the gut, and ended up seeing its profound systemic effect on neurological function, disease, even cancer.
The gut microbiome is essentially controlling the quality assurance of our most foundational biological process.
It really is.
And that control immediately opens up new therapeutic possibilities. We have strong evidence linking Q deficiency to neurodegeneration, and we have evidence that inhibiting the Q modification enzyme, QTRT1, slows tumor growth.
So that leads to a huge question.
It does. Given this complex dual role, we have to ask, what are the clinical implications of intentionally modulating the gut microbiome supply of QN or directly targeting the QTRT1 enzyme? When treating one disease like a tumor could potentially compromise a process needed to stave off another, like Parkinson's. That microbiome translatome axis is not just interesting, it is the critical double-edged sword of future medicine.